Good morning, friends. In this today's session, we will go through what is microbiology. Microbiology, before we study microbiology, we need to understand what is microorganisms are. See, microorganisms are microscopic in size and too small to be seen with the naked eyes. They are only seen with the help of microscope. And before we understand what is microorganism, we need to understand what is prokaryotic cell and what is eukaryotic cell. See, as the name suggests, prokaryotic. Pro means against, karyotic means life. So, against life cells. So, these are the cells of the bacteria. And eukaryotic cells are cells of a human cells or animal cells. Okay. Microorganisms are classified with the various methods. One of the method is depending upon what kind of organisms are there, like bacteria, fungi, protozoa, virus, and rickettsia. Examples of bacteria are like Staphylococci, Streptococci. Fungi examples like Candida. Protozoa example is Entamoeba histolytica and Trichomonas. Viruses like poliovirus, smallpox virus, chickenpox virus. And the another class is Rickettsia. What is this Rickettsia? The Rickettsia are having a diverse collection of obligately intracellular gram-negative bacteria, mostly found in ticks, lice, fleas, mites, chiggers, and mammals. Rickettsia are a disease-causing organism that resembles bacteria but multiply like viruses. They enter human from the bites of insects such as fleas and lice. They can cause disease such as typhus and Rocky Mountain spotted fever. They are also called as zoonotic pathogens. What is zoonotic pathogens are? Zoonotic pathogens refers to the pathogens naturally transmitted between animals and humans. Few examples such as animal flu, anthrax, bird flu, bovine tuberculosis, cat scratch fever. These are some of the examples of zoonotic pathogens. Now we have to also understand what is the structure of the bacteria. Here we have shown the image of a bacterium, that is a single bacteria. See, each bacterium is having a specific structure. They are unicellular in structure. They have a plasmid membrane which is surrounded by cell wall, and cell wall is also surrounded by capsule. Now each bacterium has a bacterial flagellum, that is a tail-like structure which help in the movement of the bacterium. Apart from this, they are also having small thread-like structures on entire cell. They are called as pilus. Pilus actually helps them in attachment with the host cells as well as sexual reproduction. Now we'll see the function of each layer. Capsules. Capsules offers a protective layer around the bacteria. Cell wall is a rigid layer that gives the bacteria protection and its shape. Plasma membrane is a semi-permeable layer, regulates flow of material in and out of cell. Bacteria has DNA, that is a storehouse for genetic information. Apart from this, bacteria also has cytoplasm, plasmid, ribosome, pilus, and flagellum. We have seen the function of pilus that helps in adhesion to surface and sexual reproduction, and flagellum helps in moving around in the environment. Another example is another type of classification is depending upon the shape of the bacteria. See, if they are round shape, they are called as cocci. If they are rod shape or a cylindrical shape, they are called as bacilli. If the bacteria are comma shape, they are called as vibrio. And if they are spring shape, they are called as spirally. And if the spring shape but tightly coiled, they are called as spiral. The another type of classification is depending upon the cell arrangements. That means how they are arranged together. Here we have taken one example of coccus, and as you all know, coccus is a round-shaped bacterium. So when these two cells are placed together, They are called as diplococci. Diplo, di means two. Diplococci, two cocci cells together. Classical example is Neisseria. When such bacterium is arranged in a chain, they are called as a chain type of cocci, and classical example is Streptococcus. So depending upon their cell arrangement, two chain, or if they are in a group of four, they are called as tetrad. If they are in the cluster, they are called as a cluster type of a bacterium. Tetrad are normally seen with the Sarcina, and cluster arrangement is normally seen with the Staphylococcus. Another classical example is depending upon the size or the shape. If they are very big in size or they are in a small in size. Classical example, the biggest bacterium is Mycobacterium. Mycobacterium size is usually 2 to 4 microns. Both are a rod-shaped bacteria. This Mycobacterium is a rod-shaped bacilli, and the smallest size is uh, Sira- Sriracha marcescens, which are having the size of zero point 0.5 to zero point 0.8 microns. So they are the smallest size of bacterium available. Now, the another classification is done on the basis of the gram staining. Gram positive bacteria after staining will show the purple color, and the gram negative bacteria after staining will show the pink in color. Now, why this difference comes? Let us understand. Now, look at this image. On the left hand side, gram positive bacterium is having a thick peptidoglycan layer, and that is why it absorbs more quantity of purple dye and it shows a purple color. Whereas in gram negative bacterium, there is very thin layer of peptidoglycan, but this outer membrane absorbs more of a pink color, and that is why it shows pink in color. So this is the difference why gram positive and gram negative bacterium shows different stain after the staining process is done. Now, again, bacteria are also classified depending upon their survival conditions. For examples, there are certain kind of bacteria who require oxygen for the survival. Examples are Staphylococcus species, Streptococcus, Enterobacteriaceae, Mycobacterium tuberculosis, and Pseudomonas aeruginosa. There are another class of bacteria who doesn't require oxygen. They can survive in the absence of oxygen also, and they are called as anaerobes. Anaerobes classical examples are Clostridium tetani, which causes tetanus, Clostridium botulinum, which causes food poisoning, E. coli, Klebsiella, and Actinomyces. Facultative kind of bacteria, they can survive both the environments. Like if the oxygen is there, then also they can survive. In and oxygen is not there, then also they can survive. Example is E. coli. And obligate bacteria, they are very strict. They either require oxygen or they may not require oxygen. So obligates are aerobic as well as obligates are anaerobic. Classical example of aerobic obligates are Mycobacterium tuberculosis. Now, so depending upon their gram positive staining, gram negative staining, they are further classified as a obligate, facultative, gram negative obligate, gram negative facultative. So further classification as follows. Now, there are certain class of bacteria which produces synergy. An aerobic infection may create oxygen-free zone, which build a favorable environment for anaerobic growth. For example, if the aerobic bacteria are there, they will utilize all the oxygen available there. So the oxygen will be depleted. So what will happen? They will create a new environment, oxygen depleted environment, so that anaerobic bacteria will come and strive over there. Or it can happen vice versa. So there is a synergy between aerobes as well as anaerobes. Aerobes continue to survive in spite of less oxygen because they are mostly facultative organisms. Now, another classification is depending upon their utilization. whether this bacteria is useful or it is a harmful bacteria. See, classical example is lactobacillus. Lactobacillus or lactic acid helps in the fermentation of milk to create cheese, sour cream, yogurt, and similar food that can help our gut to digest. So, humans have utilized this bacteria in the favorable condition. They have used this lactobacillus for fermentation process such as for brewing, baking, and making cheese and butter. Chemical manufacturing, they have used this bacteria for the production of ethanol, acetone, organic acids, enzymes, and perfumes. And pharmaceutical industries also use this bacteria for the manufacturing of antibiotics, vaccines, and steroids. So, depending upon their usage, these bacteria are classified. Now, these are the certain commonly found bacteria in the particular part of the human body. For example, there are millions of bacteria found in the oral cavity, but some of the examples are given over here is Peptococcus, Peptostreptococcus, Actinomyces. Another example, the commonly found bacteria on the skin is Staphylococcus aureus, Streptococcus pyogenes, Streptococcus epidermidis. These are the certain common bacteria that are always found on the skin surface. The most common infection is upper respiratory tract infection and the lower respiratory tract infection. If you look at the commonest pathogen in this upper respiratory as well as lower respiratory tract infection are Streptococcus pneumonia, Haemophilus influenza, Klebsiella pneumonia. These are the certain bacteria that are commonly found in upper respiratory as well as lower respiratory tract infection. The commonest bacteria found in UTI is E. coli, then Klebsiella, then Enterococcus group of bacteria. These are the certain sites where you will find most common type of